So hello everyone, welcome to the online course on pelagic imaging organized by Itapina, a project that aims to uh, provide opportunities for interaction of people interested in pelagic imaging across the Atlantic. We have today Savannah Judge and Harry Nelson from Yokogawa Fluid Imaging. They will talk about the FlowCAM and its applications in marine sciences. We are recording the session, and all sessions belonging to this course will be put available online with some delay, perhaps a week or 1 week delay, but they will be available online. So don't worry if you miss one of them. We welcome you to participate in all the sessions. I hope you will be able to make it. And I would like to ask Savannah and Harry to introduce themselves to the audience. And after your talk, we will open for questions, okay?
Sounds good.
Please feel free to start.
Thank you. And just to confirm, Rubens, forgive me, are you seeing my PowerPoint in presentation mode or just the title slide?
No, it's in the presentation mode.
Oh, okay. That's okay. We'll go back. Okay. All right. I'm going to start with introductions.
Great. Yeah. So hey, this is really fun. Rubens, I want to thank you for inviting Savannah and me to give a talk about the FlowCAM, and just for setting up this entire this class of pelagic imaging. We're really excited to do this. My name is Harry Nelson, and I work at Yokogawa Fluid Imaging Technologies. I have been for many years. I'm going to say a little bit more about that in a second. My background, as an undergraduate, I did major in environmental science. I got one of the first environmental science degrees ever granted in the United States. It was almost 50 years ago, but I'm very proud of that. And then I went to graduate school for business, and I've been working in business, mostly in marine science for the last 20 years, the last 17 with fluid imaging technologies, with Yokogawa Fluid Imaging Technologies.
And I'm Savannah Judge. Nice to see everybody. Well, sort of see everybody. I work with Harry. We work on the aquatics team here at Fluid Imaging, and we'll talk a little bit about what that means in the context of our company in a little bit. But I've been with the company for about 2.5 years now. And prior to starting here, I worked at the Gulf of Maine Research Institute here in the Portland area. So we're based in northern New England, about 2.5 hours north of Boston in the northeast corner of the United States. And I think with that, we'll just give a brief overview of our agenda for today's session.
Yep. So we just talked a little bit about who we are. I want to give a little bit of background on the FlowCAM. Mark really got into it the other day, last week. I'm going to delve a little bit deeper into the product development, into the company development, a little bit of the history of the company. We'll talk about the product line of the different FlowCAMs. That's the only instrument that our company makes. It's just the FlowCAM. We'll talk about the instrument itself, how we operate it, a little bit about the hardware. Then we'll talk about the software, how you can use the software to help you identify and make sense of all the data, all the images that the FlowCAM collects. And then we'll talk at length about the different applications and many of the studies that people are using the FlowCAM for when they're looking at marine science using the instrument. And for me personally, move the page down. That's me there. Today, 17 years ago, I began working for Fluid Imaging Technologies. And for me, this is a big week because on Friday will be my last day. I'm retiring after working here for 17 years. And for the work that I've been doing for the 17 years, it has been so rewarding, so personally fulfilling. I'm very proud of what we did to build a company from nothing into a company that recently was sold to a big Japanese company. But the thing that, and I get emotional about this, is working with so many cool scientists, including that guy right there, Rubens. This is a picture of us at the Zooplankton Production Symposium when it was in Pucón, Chile. It was without a doubt the most enjoyable week I've ever had in my 50 years of working. It was just so much fun. So for all of you students out there, you're going to be a part of this wonderful community of marine scientists that circumnavigate the entire globe. It's just so much fun. And as you see here, when you go to conferences like this, you develop these wonderful relationships. So, you know, for me personally, this is a big day. It's what a really cool way for me to sort of end my career with fluid imaging, to talk with you guys, and you know, to be with a good friend of mine, Rubens Lopez, who I've known for many, many years. So just a little personal point I wanted to make. I'll talk a little bit now just about the FlowCAM and how we founded the FlowCAM, the development. And really, the two guys on the left and right side of this slide here, Charlie Jensch, he was the founder of Bigelow Laboratories. He was at Woods Hole. He came up to Maine in the 70s and to start the Bigelow Laboratory for Ocean Sciences. One of his top scientists then was Mike Saraki. At the time, he's now program manager of biological oceanography with the National Science Foundation down in Washington, but he worked at Bigelow for about 20 years. And the two of them, Mike has a very strong background, both in biological oceanography, especially in marine microbes, but also in imaging that he did when he was a graduate teaching at the University of Rhode Island Graduate School of Oceanography shortly after he got his PhD. And between Mike and Charlie, you know, they had an idea of let's take the principles of flow cytometry and see if we can add imaging to it. Obviously, you get all sorts, you know, the flow cytometer, as many of you know, is really good for looking at smaller things, but you're not doing any imaging. You're construing all the information from the data you're collecting based on fluorescence, and they wanted to bring in the idea of imaging. So Mike invited his cousin, Chris Saraki, who at the time was getting his PhD at Dartmouth. And Chris is an imaging guy. He was an optical physicist. So they brought him to Bigelow, and Chris sort of took that giant leap of faith and joined Bigelow, and they wanted to see if they could develop some sort of an imaging device to look at phytoplankton. And what they wanted to do was look at the size spectrum from around 10 microns up to a millimeter, thinking that for science and for collecting data and information on phytoplankton, the flow cytometer does a good job at the small particle size range under 20 microns. They wanted to come up with, see if they could develop an instrument that could collect data up to a millimeter in size. So that would include, of course, phytoplankton and then microzooplankton. So this was in 1995. They bring Chris to Bigelow, and I'm kind of want to go just backtrack just a little bit. When we were in Mark's talk last week, I believe it was Marco Corrales, who's I think with NOAA. I don't know, Marco, if you're in the class today, but Marco asked the question, okay, somebody's got an idea to do some sort of an imaging device. And as Mark showed last week, many people have had these ideas. Many instruments have been developed, but really only a few have been commercialized where people are actually being used, where the instruments are being used in the world of science to collect meaningful and reliable and repeatable data. So that's a little bit about what I'm going to talk about here in the next few slides. But right now, this portion is where the folks were where Mike and Chris, they just cobbled together a lot of different resources and really not spending much money at all, but starting to build a prototype of what they eventually called the FlowCAM. So they took a little bit of money. They took a fluorescent microscope, and then Chris would make a lot of trips to Radio Shack at Boothbay Harbor. And then they were actually building it in a garage on the Bigelow campus in West Boothbay. And what Chris came up with was what he called the Bigelow Inflow Imaging and Analyzing System, or the BIFPIAS. Obviously, Chris didn't have a good idea as far as marketing. How are you going to market an instrument called that? But essentially, what the components of the FlowCAM was in the beginning was just a flow cell. They took 2 microscope slides and epoxied them together. They used a mercury lamp for fluorescence excitation. They had a Vidicon tube camera, and then a Gateway 400 computer. Now this was back in 1995. So here is a picture of the prototype of the initial FlowCAM. So there is the Gateway computer. That's the first computer my family ever had. There's the camera right there. You can see it's actually set up on a microscope where there will be a lens. You can see the turret there with the different lenses. And they actually sucked the sample through the microscope slide and then imaged images of the phytoplankton of the samples that they were running while the FlowCAM was at sea. And again, this is in 1997. So with that in mind, they decided, hey, let's go for an NSF grant. And they did get a 3-year grant from NSF for instrumentation, $340,000. They had a little bit of outside funding. And essentially what they came up with was an instrument, what they called the FlowCAM, which stands for flow cytometer and microscope. So there is the first FlowCAM that was ever sold. And that was to Suzanne Strom at University of Western Washington. And then this is really the second one you see here in the Siraki lab, what we call the FlowCAM, the coffin model. So inside are all the instrumentation, the components, but that was really the first commercial FlowCAM, the first model that Chris, when he started the company, went on the road and started selling. And that instrument actually is still being used by Nicole Polton at Bigelow Laboratories today. She does have a newer FlowCAM, but she uses them both. These are some of the initial images of the FlowCAM, all black and white, a little fuzzy, not so great. They've gotten a lot better. And these are some of the early models. I showed you what we call the coffin just a second ago. This is called the B2, the model of the instrument that we built from around 2006 to 2010. We also had a portable model that we sold until about 2010. We really didn't like the way it performed in the field, so we stopped making that. And then this is the model that what we call the VS, that really brought us once the company started moving along, that we built and used and sold up until 2016. I am going to talk. We also in around 2007. Or no, 2008 or 9, we joined forces with Battelle Labs. Battelle does a lot of work in the world of marine science, and they wanted to have take the FlowCAM and make it an in situ device. They do have a FlowCAM that they use at one of their labs in Washington, out in Seguin. But they came to us and said, hey, we'd like to see if we could develop an in situ FlowCAM together. So we actually did develop a submersible FlowCAM. There you see it at the top. That's the housing of it. There I am. I'm actually running it. We did a sea trial down in Duxbury, Mass. This is in 2012. And then we deployed it for about 66 days, a little over 2 months, back in 2012 at the Ponce Inlet in Florida. And, the instrument really worked pretty well, but as you guys are going to hear in a couple of weeks, Lisa Campbell is going to be talking about the imaging flow cytobot. And when it came from a business standpoint, we realized that it was going to be so expensive for us to commercialize this that we abandoned the whole idea. We love the idea of doing it, but this is one of those examples where when we sort of looked at the dollars and cents of the idea of bringing this to market, we decided to abandon it. You're going to learn a lot about the imaging flow cytobot in 2 weeks. It's a great product, and that product does a much better job than I think we would ever have done had we commercialized this and brought it to the market for in situ imaging. And so at this time, going back to 2006, that's around when I joined the company. We worked out of Chris's house. But getting back to Marco's question, we did have some funding. If it wasn't for the funding from NSF and also from the state of Maine, we really wouldn't have been able to hire people and pay people to make a company that would sell this FlowCAM successfully. But we really were cheap about everything. We worked out of a farmhouse that Chris owned, really close to Bigelow Labs. We only had 5 full-time employees. We had a lot of contract employees because we really couldn't afford to pay anybody very much. But one thing I do want to do, we had some key employees, and one being Nicole Polton, who at the time, she's now a senior research scientist at Bigelow. At the time, she was only a part-time scientist with Bigelow and part-time with us. But she's the one that would tell us what we should do and what we shouldn't be doing. And she really helped build our company and guide our company and give the guidance that we really needed to help eventually be successful. So today, that was 2000. I joined the company in 2005. We started in 1999. But today, our company is known as Yokogawa Fluid Imaging Technologies. We've got 35 full-time employees, and we will soon have, I think, 3 or 4 more. So we'll be close to 40. We work in a real office. Everybody's paid a lot better than we were back then. And then 2 years ago, about 2 years and a little over 2 years ago, we were sold. Chris and the founder of the company, Ken Peterson, they sold the company to Yokogawa Electric, a big company with worldwide presence in Japan. And what's really it's been interesting, for me, an old timer that started in a farmhouse, I was a little bit concerned. We're going to be a big part of a big corporation, but we are still our very own company. We run everything ourselves. And what is nice is we do have some resources coming from the parent company because they really want to invest and help grow the FlowCAM so it can serve more markets and actually do a better job in the instrumentation itself. And what this picture is showing, and this is where we're getting some funding and support from Yokogawa, is we're finally bringing artificial intelligence into our software. We're not really going to talk too much about that today, but that's the next product that we'll be introducing that will help folks like you make better use of the FlowCAM as far as the data goes with image recognition. And just that, so what has been really gratifying, we've sold over 1,200 of these things. We're in about 60 countries. We just added Slovenia last week to the National Institute of Biology, but we do have them in every continent. There's also one down in Antarctica. There's also a few that float around on different ships like Tara, but it's been gratifying to see these instruments go all over the world. Okay, do you want to talk about the different? So I'm going to turn it over to Savannah, and she's going to talk about the different instruments and some of the other markets that we work in.
Yeah, so this is a quick overview of FlowCAM's current product line. So Harry talked a lot about the history of the product development and how we got to where we are today. The instruments pictured here are the ones that are most commonly used in marine science and other aquatic applications. So the one in the upper left-hand corner is the FlowCAM 8000. So that's the one that you'll most often see. It includes a couple different models, the FlowCAM 8100, which just has auto imaging mode, and we'll talk more about what that means later on. And then the FlowCAM 8400, which has the option to use fluorescence triggering as well. So this is a quick overview, like I said, of the products that are most commonly used in aquatic applications today. Here are some example images collected from the FlowCAM using different magnification and flow cell combinations, just to give you a taste of the type of images that the FlowCAM acquires. So starting with the highest magnification that we have on the FlowCAM 8000, which is the 20x, image here, Karenia brevis cells. Next, we have some images collected at the next lowest magnification, which is 10x. And this is the one that's probably most commonly used for microplankton analyses in the ocean environment. For example, this collage was collected from the Gulf of Maine back in December of 2018. You have a variety of different types of organisms here, and we'll talk more about how the software works later on. But again, just to give you a taste of the type of images and what they look like at different magnifications. And notice the scale bar in the lower right-hand corner. It's probably difficult to see on the shared screen, but the scale bar is 50 microns. The next magnification, moving down or up in the size spectrum, we have 4x. So this is used to target particles in about the 30 to 300 micron range. So if you're able to make it out, the images below each of these images here represents the area-based diameter. Again, something we'll touch on in more detail later on, but we're sorting the images from largest to smallest, and you can see our largest polychaete larvae here is about 248 microns in diameter, all the way down to some really small dinoflagellates at about 30 microns. Then lastly, we also have the lowest magnification for the FlowCAM 8000, which is the 2x. So this is the one that's most commonly used for microzooplankton analyses. This particular sample was collected by the Institute of Marine Research at a fjord in Norway, looking at salmon lice. Correct.
Yep.
And then lastly, we also have another instrument called the FlowCAM Macro. If you recall, actually going back to the overview of our instruments here, the macro is in the lower left-hand corner. It looks the same on the outside in this particular photo, but what sets the macro apart is, as the name implies, it's used for looking at much larger particles. So this particular sample, we're looking at zooplankton from, think about, thank you, the scale bar. If you all can see it, I couldn't even see it. The scale bar in the lower right-hand corner is showing 2 millimeters. What sets the macro apart, again, is looking at larger particles. It can go up to 2 millimeters or 5 millimeters, depending on the flow cell that you have installed. It also has an external peristaltic pump. So where the other FlowCAM models that are designed more for microplankton use a really precise syringe pump, which we'll demonstrate later on. The macro uses an external peristaltic pump to be able to handle larger sample volumes associated with larger particles. So thinking about our company as a whole, we just touched on some example images collected with some of the most common instruments used for aquatic sciences. But if you zoom out and take a look at our whole product line, we also have other instruments that are developed for other applications as well. So I'm just talking specifically about the ones mainly in the upper right-hand corner. We have the FlowCAM LO, which stands for light obscuration, as well as the FlowCAM Nano, which is capable of imaging particles in the 300 nanometer to 2 micron range. So kind of covering that lowest end of the size spectrum. And these instruments were developed primarily for biopharmaceutical applications. So like Harry mentioned earlier, the FlowCAM was originally developed for marine science, of course, by biological oceanographers. But very quickly, people in other fields realized the power that combining imaging with enumeration can have in their industry. So this is a quick overview of all the different areas that we're in. So in addition to different aspects of aquatic research, you'll also see that the FlowCAM is used in biopharmaceutical and pharmaceutical applications. So looking at protein aggregates and silicone oil droplets. It's also used for food and beverage, as well as materials characterization. So everything from looking at the toner in a printer to chemical analyses. I did want to touch briefly on biopharmaceuticals because this is probably the second or first yeah, it's right up there with when we look at the proportion of FlowCAMs that are used in aquatic sciences versus other applications. Biopharmaceuticals is the largest part of the business. So these are a few different areas that it's used in. So specifically things like protein therapeutics, of course, academic research, quality control, and lots of other different uses. So just to touch on, we thought it was really interesting because the FlowCAM has expanded and the company has diversified into different fields that actually touch us in unexpected ways. So thinking about biopharmaceuticals, this is defined as biological materials like proteins that are used as medicine. And specifically, the FlowCAM is used in protein therapeutics. So these are proteins that are engineered in a lab for some specific medicinal use, such as insulin. And the FlowCAM is used to help speed up the development of new drug formulations and also used for quality control. A couple of FlowCAM users in the US that are probably household names by now, if they weren't already, are groups like Pfizer. So they're running samples related to drug formulations to help understand more about COVID and the flu. It was used as part of the development of mRNA vaccines. And basically proteins that surround your cell, and your immune system learns how to recognize and fight different viruses using specific antibodies. So using imaging techniques like the FlowCAM can help scientists speed up the development of these formulations over time. Another user of the FlowCAM is a company called Protein Sciences in Connecticut. They also produce vaccines and biotherapeutics. And I thought it was really interesting. They use the FlowCAM for routine testing in their bulk drug products. So what they'll do is they'll monitor a particular drug at week 1, at week 2, and then over the course of a month after it's been in storage to look at how the particle counts and particle size changes over time under different storage conditions. So a picture's worth 1,000 words. So here's just an example of some images that are collected in the biopharmaceutical world. So the upper image here are protein particles that were imaged on day 1 of a particular experiment, like I just mentioned. And then below these 2 large ones, we have protein aggregates that were imaged after 4 weeks. So the scale bar here is about 20 microns. And one of the reasons people really appreciate the FlowCAM in this area is its ability to segment transparent particles like these protein aggregates. So that was a lot about biopharmaceuticals, but before we got too far into marine applications specifically, I just thought it was really interesting to touch on how the FlowCAM has evolved over time and the many different applications that it has. So getting it back to marine science now, we wanted to provide a little bit of an overview of FlowCAM operation, both the hardware and the software side of things. So this slide shows a couple different setups and a couple different instrument models. On the left, we have a FlowCAM that was brought to sea as part of the Gulf of Maine Ecosystem Survey. This is the FlowCAM VS. If you can see, Harry's moving his mouse around to show the actual instrument. And of course, the monitors are up above it. And then on the right-hand side, we have a FlowCAM 8000. So this is the next generation of the FlowCAM that you see on the left, basically. And this is the more traditional laboratory setup. So for those of you who aren't familiar with the FlowCAM, or specifically the FlowCAM 8000, we're going to focus on this model for the majority of the rest of the presentation because it is one of the most commonly used in marine science. To give you a quick overview of what the operation workflow looks like, first and foremost, a sample is introduced into the top of the instrument using a pipette tip. Then a syringe pump draws the sample into the flow cell. This is the glass cuvette that holds the sample as it passes in front of the optics. Third, the sample passes in front of an LED, objective lens, and digital camera. And then in the bottom image here, you can see the software crops or segments the particles or regions of interest out of the camera's field of view and saves them to a database, while also recording particle concentration and particle property data. Then last but not least, the user can post-process the data, like removing duplicates, build libraries of representative images, classify particles into different groups, and export reports. So in a little bit, we're going to show a quick video of the FlowCAM operation from start to finish so you can visualize what that looks like. But one thing that the video doesn't touch on that I think is really important is sample preparation. So we get this question a lot, how do I need to prepare my sample for use with the FlowCAM? So first and foremost, one of the things that sets the FlowCAM apart from traditional microscopy is the need for prefiltration or size fractionation. The reason for this is because the FlowCAM, unlike a traditional microscope where the operator has full control over the depth of view and the field of view, with the FlowCAM, as we try to automate image acquisition, we have to control for those factors using the flow cell. So restricting the depth of view, using a specific magnification paired with a specific flow cell. And so that by nature requires filtration or size fractionation to remove particles that are larger than the flow cell, and this helps prevent from clogging the system. It doesn't always work perfectly. You'll still get clogs sometimes, but it's a really good first pass at helping prevent that from happening. So here on the right, we have a picture of some different size Nitex mesh filters or cell strainers used to separate the sample into different size fractions to look at under different objective and flow cell combinations on the FlowCAM. Next, an important aspect of sample preparation is, of course, storage and sample handling. The FlowCAM is capable of accepting live, preserved, or stained samples. So here's an image of an organism preserved in Lugol's, for instance. It's, of course, important to remember that all of the same benefits, but also limitations of each preservation method still prevail when it comes to the FlowCAM. The images you see, like the one you see here, appear as bright field images. So even if you're working with a stained sample using fluorescence on the FlowCAM, you're not going to see an epifluorescent image, you're going to see a bright field image. So keeping that in mind when deciding what types of methods to use is really important. And then last but not least, depending on the sample concentration and or the mode of data acquisition, sample dilution and in some cases even concentration may be required. So here on the right, the FlowCAM does have a really simple ratio calculator to translate your dilutions or concentrations to your final outputs, just to save you that tiny little extra bit of math. But this is really important when it comes to using the FlowCAM in fluorescence trigger mode, which we'll talk about in more detail later on. So next, we're going to show a quick video of a FlowCAM 8400 in action. I want you guys also to think about some of these key elements as we show the video. It's about 11 or 12 minutes long, and it covers a lot of different aspects, but just to help root you in what you should be looking for. I want you to notice things like how the sample is introduced to the FlowCAM, mentions of different modes of data acquisition like auto image and trigger mode, mentions of different objective or magnification and flow cell combinations, which I touched on earlier is really important with respect to the FlowCAM. Fourth, I want you to think about the camera's field of view and what that looks like when you're operating the FlowCAM and its implications for the data the FlowCAM collects. And then lastly, we'll touch on basic image recognition and how it's based on statistics. And we'll take a look at the FlowCAM software in a more in-depth video after this as well. So what I'm going to do is I'm going to go ahead and play the video. And hopefully I might need to
Hi, my name is Harry Nelson. I work at
stop sharing my screen, share my entire screen. There we go. And there is sound to this video. So Rubens, if for any reason you don't hear the sound or the video isn't coming through as we planned, just feel free to speak up, okay?
I work.
Start at.
Today is just a little overview of how the FlowCAM 8400 works. We're here in the laboratory at Yokogawa Fluid Imaging in Scarborough. Here's some colleagues, Sean and Chris. They're looking at some other instruments here in the lab. And my colleague Dominic is filming me.
Hello.
So this is the FlowCAM 8400. When I say the 8400, it can operate in two ways of capturing data. It can run in fluorescence trigger mode. This particular instrument has a green laser. And there's two channels of fluorescence detection for this particular sample that we're going to run today, we're operating this just in auto imaging mode. So when I put the sample in, what's going to happen is the sample is put in here in the funnel. It is drawn down through a flow cell. And actually here is a flow cell right here. This particular flow cell is 100 microns deep. And we're going to be magnifying it with the 10x objective. That's where the flow cell is, right in there. There's a syringe pump that draws the sample down through the flow cell, and then it goes into a waste bin. When we're running multiple samples, we're just going to be running one sample today. We have a cleaning function and a rinsing function, so you can be running multiple samples and you can clean in between samples. Inside here is a laser, different photomultiplier tubes. There's actually 2 computers. And that's all inside the instrument. And then we don't use the laser for illuminating the light to provide light for the imaging. There's a strobe light, an LED right here. So there's the LED, there's the objective. The instrument can come with 4 different objectives. Here we're running a 10x objective and the 100 micron flow cell. If you want to be looking at smaller particles, you would do a 2x objective with a 50 micron flow cell. Larger particles, phytoplankton as well as microzooplankton, we can change this out to a 4x objective. It's matched with a 300 micron flow cell. And then for larger zooplankton, we can put a 2x objective in and we run the sample through a 1 millimeter flow cell. So effectively, if you get all 4 objectives, you can image particles from 3 microns up to 1 millimeter in size. Okay, so we're going to start to run this sample. So I'm going to close everything up. And yesterday, I did a plankton net tow right in Casco Bay, very close to where the office is. The sample is in here. So I'm going to pipette some of this sample. We're not going to average it. And when we run it with a 10x objective, it takes 6 minutes to run 1 mil. We're just going to run this for a minute or 2. But there's where my sample is. And what I'm going to do now is I'm going to prime the flow cell just very rapidly. I'm doing this in what we call a manual method. We're starting the pump. So what we're seeing here, this is the camera's field of view. The sample is being drawn down into the flow cell, and now you can it's hard to see it, but you can see some particles going through there. So that's the flow cell. It's 100 microns deep, and that's about, I think, 7 millimeters across. So we can actually get a lot of data very, very quickly. So I'm going to close this window, and now I'm going to run the sample. Actually, probably need to put just a little more sample in, because we ran it pretty fast. I don't have to worry about how much sample I put into the funnel, because the software knows exactly how much sample is being analyzed. So we're going to run this in what we call auto imaging mode. And this is, we collected the sample in Falmouth, the town next to us. And now we're going to start running it. So again, this is what the camera is seeing. It's starting to run the sample. And just give it a second. And that's the flow cell. So these are the data that we're actually collecting. So the camera is going off at about 10 frames per second. And when it sees a particle having a difference in contrast with the background, it segments the particle out of the background and saves it over here. So here we're seeing camera's field of view, and these are the data that are being collected. I'm going to zoom in here so you get a little better view of the particles. Looks like we see some Cattosaurus. You might see an egg here. Lots of filamentous diatoms. So we can run this. Again, this is running at 6 frames a second. There's some serration. And so we can run this. We can actually leave the FlowCAM alone. If we're going to run a mill, it's going to run for about 6 minutes. It shows us how much time is left. in the run for 1 mil, and also how much sample has been imaged so far. So I'm going to turn this off now, and then I'm going to show you briefly how the FlowCAM does some analysis. Okay, so we've completed the run of that one sample, and we get some summary information immediately. There's many different ways we can look at these data. I'm not going to change the different graphs, but if I right-click on here, these are all the different types of graphs and different types of information we can show. What I'm showing here is the size of the particles over the frequency in terms of particle counts. And as you would expect, we get more small particles in the sample than we do large particles. And I should mention for this particular sample, we're looking at particles as small as 10 microns up to 100 microns. And then there's some other graphs here as well. We also get very, very quickly some statistics on the sample that we just ran. So this particular sample, it captured 1,294 particle images. And what's really important, we're getting our concentration as expressed in particles per mil. So the software knows precisely how much sample was analyzed over when you're running the FlowCAM. So I'm going to open up the view window simply by doing this. And the data is presented to us from the smallest particles, which we see here. We see some very small filamentous diatoms. We also see quite a bit of detritus. And then if I go to the extreme, the other end of the size spectrum, we get some pretty big guys. And here we're seeing some nuclei. This number right underneath here represents what we call the area-based diameter. And if I put my cursor on any one of these, let's put my cursor on this right here. This looks like the larvae of a mollusk. We get all sorts of information. I'm going to try to bring this guy over here. If you look here, we get information on the particle size and all sorts of parameter information, including biovolume and the size of the particle, what have you. So I'll deselect that. We'll keep looking at, this looks like this could possibly be some microplastic. And we're looking at the larger particle sizes. So what I'm going to do now is just show, and these are, right now the data is being sorted on their size. I can also sort the data based on what we call aspect ratio. Aspect ratio is defined as width divided by length. So an aspect ratio of 1 would be something that's round or square. And an aspect ratio at the other extreme would be our filaments, like you see here, the skeletonema, which is primarily skeletonema being very long and very narrow. I'm going to go back to sorting this based on their size. And we'll look at the biggest ones again. And what I want to do now is just briefly give you a quick introduction to how you can start to use the software to identify organisms of interest. And one particle we saw quite a bit of serratium. So I'm going to pick a couple of the serratium images. There's another one right now. Notice these data right now are being sorted on size, and the serratium are pretty close together in terms of their size. So I've selected the 5, here they are, and there's some statistics associated with these 5. So when I show all, and I'm going to filter on particles that look like the 5 I selected. So I click on this, and now it's just organized the entire data set on how close each image compares to those 5. Now, because we didn't have a lot of statistics, it doesn't select all the serratium that it should have, but I can do that myself very quickly. And I believe that is it. Oh, look, here, as we go through this, notice there's a dinophysis, a Hab species that I know you guys are interested in harmful algae. But this looks like the last serratium that we're seeing in the data set. I'll show what's selected. And then I can show the summary statistics for these so many particles that I've selected. And what's really important here is from this data set, It found 15, using image recognition, 15 serration images. And again, the software knows how much sample was analyzed, so we're able to get our concentration expressed in particles per mil. So the last thing I'll say is now, once we have this serration, I can save these as a library, and I'll save as a library, and I'm just going to call this serration. So the next time I run the sample, I don't have to go through this process. I can just use my serration library to compare particles in subsequent samples to the serration library. So in another video, we're going to show you more in depth of how you can use.
All right. So I'm going to stop sharing my screen. We're going to share the presentation from Harry's screen. But let's see, from current slide. Awesome.
Let's see. Where am I here? Okay. So I'm trying to find my presentation.
Yep, just do your whole screen and click share. You have to click on it and click share and then open the PowerPoint. Yep. And then play from current slide. Yep. Awesome. Fabulous. So that was a quick overview of how the FlowCAM 8400 operates specifically in auto image mode. So let's go to the next slide and just do an overview of some of the key takeaways from the video, because we covered a lot. Even though it was a pretty basic overview, it touched on some really key FlowCAM concepts that for those of you who haven't used the FlowCAM before, and even those of you who have, I think it's important to touch on. So first and foremost is the sample introduction. So you probably noticed how Harry used a pipette tip at the top of the instrument, and he added the sample using just a disposable pipetter. He happened to use a 1 milliliter pipette tip, and he was running the FlowCAM using a 10x magnification, again, pretty common for microplankton analyses. But you could also substitute larger pipette tips or even screw on a syringe for processing larger sample volumes. Some users have also engineered ways to connect the FlowCAM to a ship's underway system to move closer to a continuous sampling regimen. We'll stay on sample introduction for just one more minute. So recall how Harry also mentioned that the fluid volume doesn't have to be exact when you add it to the FlowCAM. This is because the syringe pump is really precise, and the software is programmed to know the exact volume of water that it's imaged. So if you add too much sample, the FlowCAM will stop after it reaches the volume that you told it to process. Likewise, if you add too little, it will stop collecting data when it detects that it's reached an air pocket in the sample. Lastly, Harry also mentioned towards the beginning of the video the importance of priming the sample. So this is a step that's not often talked about in basic FlowCAM demonstrations, but just like with a lot of other pieces of instrumentation, priming means pulling the sample into the FlowCAM, or in the FlowCAM's case, pulling the sample into the FlowCAM so that you actually have sample in front of the camera before you start collecting data. This is actually a really common mistake among beginner users. You may start running a sample and think, oh my gosh, why am I getting all these pictures of long black lines and bubbles? And well, that's because the sample wasn't primed. It wasn't pulled in front of the camera before you started collecting data. In the video, Harry used a method called manual priming, where the operator controls the speed of the pump and manually draws that sample into the flow cell until you can see sample in front of the camera's field of, or in the camera's field of view. The FlowCAM 8000 also has a machine prime option. That's really helpful to save time in between samples when you're running a large number of samples on a given day, and or when you're working with low sample volumes and you really need to conserve that sample and you don't have extra sample to play around with respect to priming. So next, I want to highlight modes of data acquisition. So Harry mentioned that the FlowCAM he was running, the FlowCAM 8400, has two modes of data acquisition. The first mode is called auto image mode. And this mode utilizes a continuous flow and a combination of flow rate and camera frame rate combos to take pictures of the particles in the camera's field of view independent of pigment or fluorescence. So this is, what's important to note is that this is the mode that all FlowCAMs have. So if you're using the FlowCAM, it's really important that you understand what auto image mode means, how the data is acquired, and the impact that your fluid and camera settings have on the data that you're acquiring. This mode of data acquisition is great for anyone that wants to image all particle types, that includes heterotrophs, non-living particles, independent of fluorescence. But like Harry pointed out in the video, the FlowCAM 8400 specifically also has this secondary mode of data acquisition, which is called trigger mode. The trigger mode utilizes the FlowCAM's onboard laser, and in this case, the laser is used pretty much as a mechanism to trigger the camera, hence the name trigger mode, to take a picture if a particle gets excited by the laser and emits light at a wavelength that meets one of the filter criteria on the FlowCAM. So this mode is ideal for people that are targeting specifically phytoplankton or cyanobacteria that contain pigments like chlorophyll and or phycocyanin. And other laser options can also be used to pair with specific stains, for example, using FDA to assess cell viability, for instance. And we'll talk more about trigger mode and its advantages and limitations in a little bit, but that was a key concept in the video. That data set, or that sample that Harry processed, was processed using auto image mode. So next, I wanted to highlight the importance of the objective or magnification and flow cell combinations. So recall that in the video, Harry used actually a 10x objective with that yellow stripe paired with a 100 micron field of view flow cell. So this combination is used to target a particle size range from about 10 microns up to 100 microns. As discussed before the video, working with natural samples would also require pre-filtration of anything over 100 microns to avoid clogging that flow cell. You'll still see particles that are larger than that sneak by, but this helps prevent clogging. Other objective and flow cell combinations can be used to target different size ranges, like you can use a 20x to look at 5 to 50 micron diameter cells. You could use a 4x to look at about 30 to 300, or even up to a millimeter. And again, this is just using the FlowCAM 8000 family, so the FlowCAM 8100 or the 8400. Next, I wanted to just touch on the camera's field of view. So if you remember from the video, when Harry initiated a run, we got to look at the live view of what's happening in front of the camera. So in actuality, the FlowCAM's LED light is flashing really, really quickly, like a strobe light, to help reduce image blur. It's kind of like if you were in a dark nightclub with the strobe lights flashing and you look around and everyone is kind of flash freezing like this. It's kind of a similar concept when you think about it, oversimplification, but it's the same with plankton. So all FlowCAM 8000 models use this technique to take pictures of the entire field of view and then segment particles out of the field of view. What's also important to note is that all FlowCAM models of this family, of this type, all come with field of view flow cells as the standard. So people that are using older versions of the FlowCAM, like the FlowCAM VS, and even some of the older ones that Harry mentioned before, sometimes you had the option to choose whether you wanted a field of view flow cell or what's called an extruded flow cell, meaning that you're not imaging the entire width of the camera's field of view. So this is really important, and we ultimately made it the standard because in a tool where you're essentially using a camera to subsample from a larger sample, being able to image the entire width of the flow cell allows you to see and image a larger proportion of your total sample and achieve higher efficiencies. Lastly, at the very end of the video, Harry gave a quick overview of how to pull similar looking organisms out of a sample data set using basic image recognition. And I really want to emphasize here that the FlowCAM's image recognition capabilities right now are based on statistics. So what this means is that for every particle you select that looks similar to the other ones, The software is comparing statistics like the mean, the minimum, the maximum, and the standard deviation for each of many, many different particle properties, such as diameter, color, aspect ratio, and others. So what this translates to in practice is that the image recognition performs best with particle selections that are unique from everything else in the sample and also uniform compared to each other. So Harry used the example of serratium, which is now called tripo since the video was made. And this is a really unique looking organism. Like he pointed out, they're all kind of similar in size. They have a really unique morphology that differentiates them from other types of particles in the sample. And something like this will perform really well with image recognition that's based on statistics. What visual spreadsheet struggles more with is organisms that are really similar to each other morphologically. So think of diatom chains that all look like long, skinny filaments, like your skeletonema, leptocylindrus, or things that vary significantly within their group. So again, chain-forming diatoms being one of them. You could have a single cell, or you could have a chain of 8 cells. They're the same organism, but the FlowCAM would have difficulty putting those together in the same group. So that means you may need multiple libraries to target different morphologies. So organisms like those will just require more fine tuning because they get confused with one another or may need different representative libraries to target. Awesome. So next, I just wanted to show a quick overview of the different objective and flow cell combinations, because we've talked about this a lot so far. It's one of the really neat things about the FlowCAM is that, at least with the FlowCAM 8000, you have the ability to switch objective and flow cell combinations in order to target different size ranges. Here's what the box would look like. This one's pretty used. This is one of our FlowCAMs. But on the right-hand side, you see 3 of the 4 different objectives, the 20, the 10, and the 4. And there's also a 2x not pictured. And then their corresponding flow cells and also syringes. We have different syringe pump sizes to correspond with each of these combinations to ensure the accuracy of the flow. So if we go back to the objective matrix for 1 second, you can see these different combinations. The field of view flow cell size in microns represents the depth of the flow cell at its narrowest point. So you will see organisms that are larger than this number, assuming that you pre-filtered the sample, depending on the angle that it passes in front of the camera at. But this is a really important guide when thinking about whether or not the FlowCAM is appropriate for a particular application or research project, because it's so highly driven by size. So now we can move on to, I also wanted to touch on continuous flow, because this is another thing that really sets the FlowCAM apart from other types of technologies. Continuous flow, meaning that the sample is passing through the FlowCAM at a preset rate. It doesn't use sheath fluid. We're just looking at the sample itself. And it has some advantages and some distinct disadvantages. So one of the advantages being, of course, you can look at a broad particle size range, like we've just talked about in the context of the objectives. You can also change the sample processing speed. So as long as it falls within the recommended range of flow rate and auto image rate, as long as those 2 variables are paired together appropriately, you have some flexibility in terms of the sample processing speed. And then lastly, you also have, like we talked about earlier, multiple modes of data acquisition, specifically auto image mode or trigger mode. But on the flip side of that, continuous flow does have some disadvantages. So broad particle size range, that lives actually in both camps, because while it's great to be able to target different size ranges, on the flip side, sometimes combining the data and looking at different size ranges can be quite challenging. For example, we talked about the importance of libraries, building representative images. You may need different libraries, or you will need different libraries for different particle size ranges, and that adds time to the overall sample processing. Next, as we've touched on, flow cell clogging, that's part and parcel, and the need for sample fractionation. And, that doesn't add a whole lot of time, but it adds a really important step that people don't often talk about, especially in the published, in the literature. And then lastly, sample dilution. This is particularly important for trigger mode, which we're going to talk about in more detail in the coming slides. But the FlowCAM basically is simulating a flow cytometer in the sense that we need to make sure that only on average about one particle is in the camera's field of view at a time when an organism triggers the laser to take a picture. Otherwise, those organisms will be assigned the same fluorescence values and make differentiating between groups based on fluorescence more challenging. Did you want to talk about this slide?
Sure. So I'm going to talk a little bit about just kind of the design of the FlowCAM and the FlowCAM's architecture. So you see the schematic on the left. I did mention this a little bit in the video, but there's a laser, there's a camera, and for the FlowCAM 8400, it's got 2 photomultiplier tubes that picks up the fluorescence information. And when we're doing that, the instrument also has 2 objectives. One objective is between the LED and the camera, and that's for illumination, for imaging. The other objective is to focus the fluorescence information, the photons, as they are released through the PMTs and then picked up by the PMTs. So the FlowCAM can have one of 3 lasers. You get people saying, oh, can we have all 3 lasers? Well, unfortunately, no. You have to decide between a 488, a 532, or a 633. And in all of the instruments of the 8400s, channel 1, that PMT is picking up chlorophyll, any emission that's greater than 650 nanometers, with the exception of the FlowCAM Cyano. The FlowCAM Cyano has a 633 laser. It'll excite chlorophyll as well as phycocyanin. And then with channel 2, we have 3 different bandpass filters that we're picking up fluorescence. With the 488 laser, we're picking up green fluorescence. And that fluorescence is typically, people will be using that, the blue laser, which will excite chlorophyll, as well as the various FITSI stains, a green fluorochrome. So Savannah mentioned that FDA, and there's some other different types of fish probes that will be excited by blue light. The standard FlowCAM for marine science has the green laser, 532 nanometer, and in channel 2 we'll pick up the phycoerythrin signal. It's a bandpass of 575 nanometers plus or minus 30. And then with the FlowCAM Cyano, it has a red laser, and in channel 2 we'll pick up the phycocyanin signal. So that's essentially the architecture and the fluorescence information that we're getting with the FlowCAM 8400. So, you want to talk about the trigger mode and auto imaging mode?
Yeah. So, as we've talked about a few different times so far, the FlowCAM 8400, this is the FlowCAM that has the laser option or the choice of 3 lasers, has these 2 different modes of data acquisition. And we just wanted to give some visuals and touch on, why you might use trigger mode versus auto image mode and what some of the benefits and limitations are. So here in this image, we just did a quick sort of scan of a sample collected from the Gulf of Maine, where we've highlighted particles of detritus with yellow dots. So auto image mode, if you recall back from our key takeaways from the demonstration video, is imaging all particle types independent of fluorescence. So you're going to get a lot more detritus, but you're also, on the flip side, going to be able to see heterotrophic organisms. And if you're interested in particulates, non-living particles as well. So depending on the research questions being asked, this mode of data acquisition may be the most appropriate. On the other side of the slide, we have trigger mode. And again, this is like Harry just described, when you're activating the laser and using the laser as a mechanism to trigger the camera to take a picture of the field of view based on fluorescence. So here you can see we have a lot fewer particles, detrital material. You will still get some, especially if the concentration of your sample is a little bit too high. That's where sample dilution as a sample preparation step comes into play. But that's just a quick overview of auto image and trigger mode. And if we move on to the next slide, Harry also mentioned the option of assessing cell viability using specific stains. And if this is an application that interests you, this is where it's really important to make sure you're identifying a FlowCAM laser with an appropriate wavelength and filters, making sure that it's paired appropriately with the stain that you wish to use. I'll pass it back to you.
Yep. And as I think many of you folks that are listening, mostly you guys are marine researchers or students of marine science. But these are that's why the FlowCAM was developed, as going back to the earlier part of the class. But these are a number of the different applications that people are using the FlowCAM for in our aquatic applications. The main one, of course, being marine research and freshwater research. Also, marine harmful algal bloom monitoring and freshwater HAB monitoring. We do have a lot of folks using the FlowCAM in aquaculture. I'm going to drop down to the bottom two bullets in algae cultivation, whether you're doing large scale algae cultivation, like you see there, that's Cyanotech, a company out in Hawaii, or also doing just algae cultivation just in a small lab, culturing algae like they do at Bigelow or like they do at the Texas, University of Texas algae cultivation lab. And then, of course, with sediment analysis, and there really are quite a few other applications in the aquatic world. I do want to focus a little bit on the drinking water application. So we have a lot of companies, a lot of drinking water companies, mostly here in the United States, that are using the FlowCAM specifically looking for taste and odor algae that when the cells lyse, they'll produce different types of metabolites like MIB or geosmin, which will give an off taste to the drinking water, and also in drinking water world, drinking water utilities using the FlowCAM to look for cyanobacteria, which of course can produce toxins, and you don't want that in your drinking water. And the way that the FlowCAM will work on this is you'll be using the instrument that has a laser, a red laser, and this is picking up the phycocyanin signal. And what's pretty cool, remember going back to what I was talking about earlier, we have two channels of fluorescence information. Channel 2 has chlorophyll. Channel 1 is with the red laser is phycocyanin. And what's pretty cool with the way that the FlowCAM works when we're looking at the fluorescence information, not the image, but the information provided of the parameters on channel 1 and channel 2 fluorescence, they're pretty much in equal ratios when you're looking at cyanobacteria. So what you're seeing there, that screen on the left, it's a scatter plot showing the ratio of channel 1 to channel 2 fluorescence. And that one line at the 45-degree angle where the ratio is actually pretty close in 1:1, that's where we're going to see cyanobacteria. When the ratio is a little bit higher, it's going to have more chlorophyll, and that's going to show mostly the diatoms and other algae. And then when the ratio is a lot lower, we're getting detritus and decomposing algae because there's very little phycocyanin or chlorophyll in that particular ratio. So this is where the FlowCAM equipped with in trigger mode, when you're running it in trigger mode, equipped with a red laser, it really does do a very good job of differentiating cyanobacteria from algae and then from other particles that will be in your sample. And this is why many drinking companies, utilities in the United States, a few overseas as well, will be using the fluorescence information that you're getting from the FlowCAM cyano. And this is just one example. It's just Wichita, the city of Wichita, Texas, Wichita Falls. And what's really cool, you see that in the lower right-hand corner. That's these guys are actually tasting their water to see, hey, does our drinking water have an off taste, either caused by MIB or jasmine? But we really do get a lot of folks that are, the drinking water companies don't want funny tasting water, because when the water tastes funny, their customers, you and me, will call them saying, hey, my water tastes funny, and what can you do about it? And this is where we really have sold many FlowCAMs into into public utilities, looking for either cyanobacteria, or they do have data on the different types of algae that will cause taste and odor issues once the cells start dying and start lysing.
So now we're going to
Stop sharing.
you can keep sharing for a moment.
Okay. Yeah.
Before we start the video, thanks. So we just again talked about some of the uses for fluorescence trigger mode. Most recently, Harry mentioned in the drinking water space. Now we want to kind of get back into the software and, really dive a little bit deeper into how the software works. So in the FlowCAM operation video that we showed a little bit earlier, we gave a general overview, but the goal of that video and demonstration was really to show you how the hardware operates and sort of see the workflow from start to finish. In this next video demonstration, we're going to be focusing on the software. So in the video, I want to point out a few key things. We're going to sort of structure the video into how I like to visualize it, which is 3 different levels. So level 1 is where you select similar looking particles within a single run and use the visual spreadsheet's image recognition capabilities to group together similar particles, again, within a single run. Then level 2 is when you combine representative images from various runs or multiple runs into image libraries. And then level 3 is where you convert those libraries into statistical filters, which is pretty much just a numerical representation of your image library, and then classify your samples. So that's when you take your classification templates and apply your representative images to new data sets that the FlowCAM hasn't seen before. After this, we'll again review some key takeaways, just like we did with the first video, and then touch on the use of Visual Spreadsheet with other types of data platforms. So Harry's going to stop sharing his screen, and I'm going to share mine. And we will play a video. This is going to show a similar presentation in a presentation format, but with some videos interspersed. And there will, again, be sound. This time there's also subtitles. I apologize if it looked like the last video was a little bit lagging, but hopefully it's quick enough to be able to follow along with what's happening. If not, just say something, Rubens. Thanks. Welcome to an introduction to Visual Spreadsheet and image recognition. I also do want to point out, for those of you in the room who are FlowCAM users, and I know there's actually, I was looking at the list a minute ago, there's quite a few of you. I also just want to point out that the videos we're showing today are focusing on Visual Spreadsheet 5, which is the current iteration of Visual Spreadsheet. Many people are still using Visual Spreadsheet 4. The image recognition tools haven't changed from 4 to 5, but I just want to acknowledge, for those of you who might be looking at these videos and thinking to yourself, that doesn't look familiar, just be aware that the tools in the background are the same, but the view and the look of the window and where things live has changed. So just something to keep in mind. In this video, we'll start by introducing the Visual Spreadsheet home screen, then discussing particle property data and how they're derived. Then we'll discuss image recognition tools that are built into Visual Spreadsheet. And finally, we'll classify some example data. First, we will orient you to the Visual Spreadsheet home screen. The home screen displays histograms, scatter plots, metadata, and other statistics that can be used to quickly assess your data at a glance after completing a run and before conducting further analysis. The graphs and filters on the home screen can also be used to call up the images associated with specific data points, allowing the user to quickly investigate the images behind the data. The home screen is customizable, allowing you to change which graphs to show and to remove or rearrange sections to your liking. When we open all of the images associated with a particular run, You will notice a number below each image. This is the area-based diameter for each particle. You can also sort the images by one of Visual Spreadsheet's many other particle properties. For example, aspect ratio is used to separate round particles from filamentous particles, and intensity is used to separate light transparent particles from dark ones. To understand how Visual Spreadsheet derives these data, it is important to understand what the FlowCAM sees when it records an image. To illustrate this, let's look at the binary image overlay. When the FlowCAM takes an image, it automatically segments that image from the background and creates this layer based on your context settings. Each FlowCAM is calibrated to convert the dimensions of a single pixel into microns, which is how it is able to derive the many particle properties listed here. Next, let's explore some of Visual Spreadsheet's built-in image recognition tools. Visual Spreadsheet uses a statistical pattern matching algorithm to help the user select similar-looking images. Visual Spreadsheet's image recognition tools are organized into 3 levels. Level 1 involves manually selecting similar-looking particles from within a single run. Level 2 allows you to combine representative images from multiple runs into a library. And level 3 is when we can convert those libraries into filters to auto-classify our sample and check the algorithm's work. Let's begin with level 1, selecting similar images within a single run. First, we'll select 2 examples of similar-looking organisms like the diatom Pleurosigma. Each image has many particle properties associated with it, and after selecting at least 2 images, we can view the statistics for each of the particle properties, including the mean, the minimum, maximum, and standard deviation. Next, we will tell Visual Spreadsheet to review the rest of the run and pull the images that are most statistically similar to the ones that we selected up to the top. Then we can select these images and save them as a library. Next, we move on to level 2 of image recognition, which is where we use our library to identify similar organisms from other runs and add those to our library. This step allows us to strengthen the statistics of our library. First, we'll open our library in the background. Next, we'll open a new run or set of runs that we did not use to create our library. Then we'll filter our new data based on our library instead of manually selecting images as we did before. From here, we can add the new images to our library. Remember to only include similar-looking images in a library, as outliers can skew the statistics and reduce your library's performance. The more unique an organism is from other types of organisms in a sample, and the more uniform the examples in your library are to each other, the better your library will generally perform. In practice, this means that you may need more than one library for a single taxon if it has very diverse morphologies or image types. When you test your library, make note of the number below the images. This is the filter score, which indicates how closely an image matches the statistics of the images in your library. Once we test our library and are satisfied with how it performs, we can save our library as a statistical filter. A statistical filter is a numerical representation of your library and is what will allow us to combine the image recognition power of multiple libraries into a single classification later on. We can also add an additional value filter at this stage for the filter score so that our filter accepts more or fewer particles. Now that we've created a library and filter for one taxon, we would repeat the process for other taxa of interest. Focus your initial energy on the taxa that are most abundant, as rarer taxa can be classified manually until a large enough library is developed. Once we have several libraries and statistical filters created, we are ready to move on to level 3, the final level of image recognition tools in Visual Spreadsheet, the classification window. For this example, we created libraries and filters for Pleurosigma, Dinophysis, Nauplii, Skeletonema, and Tripos lineatus. The next step is to create a new classification. The classification window is what allows the user to organize their data into different classes or groups in order to export data to Excel. To initiate a classification, the first thing we will do is open a data run. The classification is specific to each run. Next, we will open the classification window and create a new classification. Then we will proceed to create a class for each of the known groups we plan to enumerate. This is where we can add the statistical filters we created from each library to enable semi-automated classification. It is important to put your best performing classes at the front of the classification. The classification window works like a stack of sieves, pulling out images that match each filter as the images move through the series. So it helps to put your strongest filters first. Then we'll perform the auto classification operation. This is where Visual Spreadsheet does its best to sort the data into the correct class. After reviewing the initial auto classification, the next step is to fix its mistakes. This involves removing any images that were misclassified in the classification window. Then it is important to skim through the open view window to classify any images that did not get picked up by your filters. To help identify images that were not classified in the open view window, you can still utilize level 1 image recognition tools such as selecting like particles to group together similar organisms and then move them to the correct class. Continue with this process until all unclassified images of interest have been moved to their correct class. As you review the open view window for images that were not classified, you can also take the opportunity to identify additional classes that you would like to count but did not make classes for before. To do this, simply create an additional class for the taxon of interest and move the target particles into that class. Here, we have created several additional classes for organisms that were less abundant in our sample. Once you have finished classifying your sample, the final step is to export your data to Excel and save your classification template. To export to Excel, you can choose which particle properties and statistics you would like displayed in addition to count and concentration. Then, export your classification summary, save to your computer, and open. Under metadata, we see the count and concentration in particles per mil for each class, as well as summary statistics by class. Now that you've created your first classification, you can save your classification as a template to apply to future runs. This will save you from having to recreate your classes and filters each time. Go ahead and save your classification as a template and give it a name. Then we will open a new run and open a new classification using our template. You will notice all of the same classes, which we can then use to classify the next sample run. We hope that this video helped provide a good introduction to Visual Spreadsheet 5, with a focus on the image recognition tools available to help you classify your data. If you have any questions or would like to see any part of the process in more detail, please contact us for more information. All right, so I'm going to stop sharing again and pass it back to Harry's computer. But yeah, I hope that video, while it was kind of long and a little bit, repetitive, I hope it gives everyone, especially those who haven't used the FlowCAM or Visual Spreadsheet before, and even those of you who have used it, but maybe an older version of the software, insight into what the workflow generally looks like, particularly for microplankton. So I want to touch on a few key takeaways or summarize a few key takeaways from the video. So the first and foremost is that image recognition in Visual Spreadsheet is based on statistics. So you can imagine that the images in each of your image libraries is like a data point within a statistical data set. So the tighter those statistics are, the better it will perform. Another really key takeaway that you may have noticed, but I didn't specifically highlight in the video, is that it's initially up to the user to assign class labels. So the FlowCAM does come with example libraries that users can use right out of the box, but they really are just designed as that, as example libraries. It can help you get started, especially for users that are less comfortable with algae identification. But the key takeaway here is that it's still up to you, the user, to build libraries using your instrument and to assign class labels to the taxa that you're most interested in. I also want to point out, I'm not a taxonomist, but I do want to point out that, we primarily use the word genera as opposed to species level identification in the demonstration video. And that's pretty reflective of what it's actually like using the FlowCAM, using its built-in basic tools. It's not a tool that's designed for species level identification. You'll notice I did have Tripos lineatus, which is pretty unique and pretty easy to identify. But in general, you're making your decision based off of a single two-dimensional image that may be imaged at different angles, may look different during different times of the year, and you're dealing with a single bright field image. So unlike the microscope, where you have full control and you can spend as much or as little time on any particular organism as you want, with the FlowCAM, you're stuck with whatever image it grabbed from the camera's field of view, right? So for that reason, you can often get to the genus level depending on your comfort level with taxonomy, how unique an organism is compared to everything else in your sample. But don't think that it's going to get you species level data unless it's a really morphologically unique organism. Next, I want to point out that the classification process within Visual Spreadsheet is very iterative. So in the example that we showed here, we just showed some basic libraries using a handful of runs that we combined into sets from a specific series of samples. But as more images, and I didn't really show this in the demonstration, as more images are added to your library, say you're using the FlowCAM over the course of a field season, and every week you're getting new samples, and you may want to add, or you should add, additional examples of the images in your libraries to your library. So that process is really iterative, and it requires adding additional images and updating your filters and classification template. So a lot of users may, do this on a regular basis. Maybe every so many weeks, they make sure to update their libraries and see how that improves their performance. Lastly, something that wasn't shown in the video is that I think is important to note is that you can save multiple independent classifications per sample. So what I mean by that is, in this example, I just use a generic classification A for a particular sample. But what this means is that you can actually classify the sample in different ways. So you can either go back and reopen an existing classification and reorganize it. You could check somebody else's work, someone else who's maybe a novice at using the FlowCAM or at identifying phytoplankton. It also means that you could classify the same data in different ways for different projects. So maybe you have a long-term time series and someone else in your lab wants to use that data set to look at a totally different research question than you are. So that's something we didn't show in the video, but that's a really important tool within Visual Spreadsheet, is the ability to classify and reclassify samples in different ways. So we can go on to the next slide. So now we just talked a lot about the tools within Visual Spreadsheet, that's the FlowCAM's proprietary software. But we also want to transition now into talking a little bit about other data platforms that exist and other types of image data that can also be used within Visual Spreadsheet. So the first one is Ecotaxa, and I know we have several people in the room that are either users of Ecotaxa or even developers, co-developers of Ecotaxa. If you haven't heard of it, definitely check it out. It's a really amazing platform online that's used to share, annotate, classify, and archive image data. And what's especially neat about it is that it encompasses data from multiple different instruments, including not just the FlowCAM, but also the UVP, the IFCB, and others. And what this is really moving us towards is a direction where, the FlowCAM is such a localized instrument. Platforms like Ecotaxa enable data sharing and remote collaboration in ways that aren't really currently possible. And in the future, we're hoping to develop or incorporate tools into Visual Spreadsheet that make it easier to utilize and take advantage of platforms like Ecotaxa.
Okay, I'm going to talk just briefly. Last week, Mark did mention that he, with his Zovis instrument, We worked with Mark, this is quite a few years ago, and we provided Mark with the visual spreadsheet software so that he could process his Zovis data, his Zovis image data with visual spreadsheet. So we have worked with a couple of different folks over the years. Of the left-hand corner, many of you might be familiar with, if you're looking at diatoms and you clean diatoms, you digest them, and you're just left with the silicate shell, if you will, of the diatom. Folks will look at diatoms on microscope slides and identify them, count them, and measure them. Where we're able to take the FlowCAM's visual spreadsheet software, we convert, like we would do with all of these different image data sets, you convert them to an AVI file, and we're able to process the AVI files with visual spreadsheet. We've done the same, so that the diatoms that you're seeing there, they're actually, we're doing some freshwater diatoms. They're from Sarah Spalding's lab at University of Colorado in Boulder. We've done the same thing with forams, working with a scientist from the University of South Florida. And we've also been able to take IFCB data and process it with a visual spreadsheet. So let's see if I can get this, I can show you all this. And I'm going to open up visual spreadsheet. Oops, that's the wrong version. I need to do visual spreadsheet 4. And Rubens, can you see it? Can you see my slide? Can you see my screen, and you see what looks like a visual spreadsheet?
Yes, I can.
Great. Okay. So I'm going to open up. I think I'll just do 2 of these for the sake of time. What I'm going to do is we've worked with Sarah Spalding's student, Ian Bishop. Actually, he's now Susan Mendendauer student at URI, and we're able to process images from his an AVI file of a bunch of static images that he linked together in an AVI file, and we can process it with visual spreadsheet. So let me see if I can find this here. Here we go. So this is kind of what Savannah was showing you earlier in the demonstration, when we're just like we're analyzing a sample, running the sample through the FlowCAM, but now we're just running an AVI file through visual spreadsheet. And you saw this before. This is the camera getting background calibration. This is the collage file that's being collected. And this is what, in a sense, what the camera is seeing. And we're able to segment the images from the background, and we're getting a collage file like you see here. So again, this is an example of where we want to work with scientists like you guys, possibly using our software to identify images and data from either your device or different ways that you have collected image data. So this is what we were seeing earlier. Here's a presentation of the data after it's been collected. And I'm not going to go through all of this, but here are the diatom. Here are images of the diatoms that are captured from, again, from diatom slides. And you can do the same thing that we were doing before that Savannah was showing you. You know, select a few images, and you can do image recognition, finding like particles, what have you. So this is just one example using diatom slides. I'm also going to show you the same thing looking at IFCB data. Do this pretty quickly. So again, we're working with one of Paul Flakowski's grad students. I think these are data from a Southern Ocean cruise. And I'll go to the IFCB file. And again, he took the IFCB data, put it into an AVI file, and we're able to process it through the FlowCAM's visual spreadsheet. So this looks a little bit different, but what you're seeing here is those red boxes are, as we're processing the data, the image data, it's segmenting that image from out of the background. These are these tiny little dinoflagellates, and we're going to end up with a bunch of image data. Again, this is collected from the IFCB. So I'm going to shut this off, And here's the data that we collected. And it looks just like you're seeing in visual spreadsheet. We do get the particle size range. That's the scale bar that you see in the lower right-hand corner of 100 microns. Again, the whole purpose we want to do this is we know there's lots of imaging devices out there. There's lots of ways of capturing images of any small particle, and we want to be able to support scientists all around the world with different platforms with our software. So, do you want me to keep going with the presentation?
Yeah, that'd be great.
Okay, great.
Awesome. So Harry just obviously touched on the ways in which other types of data sources can be used within Visual Spreadsheet. Now for the last portion of the session, we have about 20-25 minutes left. We want to focus on specific applications and studies within marine science specifically, which is ultimately why we're all here, right, in this pelagic imaging course. And I know some of the authors of some of the papers that we're going to touch on are in the room. So please feel free, if you have additional comments that you want to add, please feel free to speak up. We welcome the interruption. This is your work. We're just presenting it in a really oversimplified manner. We're going to be touching on some key studies in just a slide or two each. So we could spend honestly all day talking about all of the different studies that have been done using the FlowCAM. So there are a few key areas that we'll focus on. Yeah, I'll get to that in just a moment. Phytoplankton, zooplankton. I also want to talk about particle transport. We'll talk about a couple examples there. And as Harry mentioned, there are also other applications in marine science that are of a more industrial nature, so like aquaculture and algae cultivation. So here on the right, we have a simplified version of the biological pump from NASA. And the FlowCAM, just like lots of other types of instrumentation or instruments, you know, you can use the FlowCAM for all different aspects of the system. So all these different components, like looking at grazing, sinking aggregates, primary productivity, so many different elements where this type of instrumentation and just imaging in general can be and is applied. So this is just an overview of some of the most recent publications from June of this year that have involved the FlowCAM in some way or referenced the FlowCAM in some way. Or I stand corrected, actually. No, we only include papers on this list that actually use the FlowCAM. There's lots of other papers that reference the FlowCAM, but these are ones that actually use the FlowCAM to collect data. So if we move on to the next slide, we want to highlight now a few key studies in some of those specific applications that I mentioned earlier. And I want to take a look at starting with Alvarez et al. 2018. So we're going to start with, I should say, phytoplankton. We're going to talk about a few key phytoplankton studies. Just to give you all a flavor for how the FlowCAM has been applied in this area before. Then we'll move on to a couple of studies featuring or focusing on zooplankton, and then one or two focusing on particle transport, and then we'll wrap up and open it up for questions. So in this particular study, it was conducted in Spain in 2018. This is a monthly monitoring program of nano and microplankton. And what was really cool is this particular study was looking at the differences between or similarities between abundance, biomass, and taxonomic diversity between the FlowCAM and traditional microscopy. And as you can imagine, this is a question that we get asked a lot, is how does the FlowCAM and other devices for that matter compare to the gold standard of traditional microscopy? And the authors reported a minimal difference. There were some key elements that are worth noting, like the effects of sample preservation and inaccuracies in automatic classification. And if you think back to the software demonstration that we saw earlier, you can imagine how iterative that process is in its current form, and the implications that it has for automatic or semi-automatic classification. There's always going to be mistakes. The software is never 100% accurate, and it takes a lot of human intervention, human effort to correct those mistakes. Another thing that's really interesting about this paper is that, unlike many others, they used both auto image and trigger mode, which we've talked about extensively in today's presentation. So if you have questions about the implications of those for data collection, I definitely encourage you to check this paper out. And ultimately, one of the key takeaways is the fact that imaging techniques like the FlowCAM and microscopy, while very different, are of course complementary techniques. So with the FlowCAM, of course, what you gain in the speed and automation of the data acquisition or the image acquisition, you lose with respect to taxonomic resolution. So as we saw in the demonstration earlier, you might be able to get down to the genus level, maybe the species level, but the tool isn't designed for that. It's ultimately, at the end of the day, a statistical tool to be able to gather a large amount of data very, very quickly. Whereas if you're really looking for species level taxonomic information, that's where the microscope still wins out, because you have full control over that depth and field of view. Oh, thanks. Next, another key phytoplankton paper that's really cool is Mendendauer et al. From 2020. And this study looked specifically at cell size and abundance using the FlowCAM, but comparing it to a lot of other different techniques beyond just the microscope. They also incorporated Coulter counters and flow cytometry. And what was interesting about this study is that they did both monocultures in the lab as well as natural plankton assemblages from the North Atlantic. So there's ultimately, one of the goals was to assess the utilities of different instruments, because like we mentioned in the last study, when we're talking about FlowCAM versus the microscope, different tools are geared towards different things and have specific strengths and weaknesses in different areas. So when one of the key outcomes of this study, when we're specifically looking at the FlowCAM, is highlighting some of those advantages but also disadvantages. So it performs really well when you want to be able to classify things in different ways. It has a large particle size range, like we talked about, through the use of multiple objectives, and the fact that the workflow is reproducible. Once you establish the settings that you're going to use for your study, which is a really, really important step that we'll talk about a little bit later, those same settings will be applied to the rest of the samples that you collect, and makes it a lot more easy for different people to work on the same project, as opposed to microscopy, which has its advantages and disadvantages. Lastly, some of the disadvantages, the image processing, as we've talked about how iterative the process is. Another key one that's important, and we get asked this question pretty frequently, is about picophytoplankton. And the FlowCAM, at least the FlowCAM 8000 family that we're really focusing on so much in today's presentation, doesn't do picophytoplankton. They're too small, even for the highest magnification of 20x. The minimum magnification, as you can see here, the authors grouped images by different size classes. So the smallest class that they presented here in this figure is the 6 to 10 micron range. So that's a really key component when you're thinking about imaging techniques like the FlowCAM, is the importance of size fractionation and the consideration of which classes are left out just by the nature of the limitations of the optics. And then lastly, the necessity of either concentrating or diluting the samples. We've touched on this before, especially in the context of using trigger mode on the FlowCAM 8400. And that's the equivalent of the FlowCAM VS4 for those of you in the room that are more familiar with the older platforms. So really, really great study. Highly recommend. Well, I can say this about all of them. Go check them out. You can read them and let us know if you have follow-up questions. The last phytoplankton study that I wanted to touch on is a long-term time series, Balch et al. From 2018. This study was published in 2018, and that's based on the Gulf of Maine North Atlantic Time Series, or NATS for short. It's an ongoing time series. Well, actually, has the time series ended?
No, it's still going on.
Oh, it's still going on.
Yep.
The transect would be from Portland, Maine to Yarmouth, Nova Scotia, which you can kind of see here from figure 1, an excerpt from figure 1. The time series utilizes a ship of opportunity. Is that right? A ferry.
Yep.
With a ferry box. to collect regular oceanographic measurements across the Gulf of Maine, which has actually been identified for those of you in other parts of the world. You know, the Gulf of Maine, our little neck of the woods here in the Northeast United States, has been identified as one of the fastest warming bodies of ocean water on Earth. So it's a really, really important area of study. And the fact that we have long-term time series like this one, like NATS, are so important, obviously. So as you can imagine, this time series collects a wide variety of ocean data. Still on the last slide. A wide variety of ocean data, including physical, chemical, biological, and biooptical measurements. So we can go to the next one. So focusing on the FlowCAM specific part of this time series, of course, it's just a very, very small part of a much larger effort. The FlowCAM was specifically used to quantify really broad classes of particulates and phytoplankton to look for broad trends and concentrations over time. So there are about 5 broad classes that have emerged from the time series. So they were looking at general particulates in different size ranges, specifically under 4 microns and then 4 to 12 microns, the ones that are really hard to visually, if not impossible to visually identify with the type of FlowCAM that they use. They also looked at coccolithophores, pinnate and centric diatoms, and naked and armored dinoflagellates. So the image on the right here shows a Hofmüller diagram that illustrates the changes in particle concentrations through space. So we have the longitude on the x-axis, and over time we have the different years. And what's interesting to notice is on the left-hand side, if you can see, we have 1, 2, 3 different segments of the time series from 2006 to 2018. And what's important to note about this is that the authors have identified 3 distinct epochs within their temperature analyses from this time series. So the Gulf of Maine, it's warming extremely fast, and this epoch number 3 from 2010 to 2018 represents the hottest of the 3 epochs, especially in the eastern Gulf of Maine. So this study, illustrates, among other things, the ability to use imaging techniques for long-term data series, but also it illustrates the importance, connected with that, of the importance of consistency with respect to configuration. So the authors made a comment that, prior to, I think, 2006 or 2008, I can't remember exactly, they didn't present data of particles under 4 microns because they were using a different flow cell and objective combination than they did for the rest of the study. So we'll talk a little bit more at the very end about the importance of settings and consistency later on, but really cool study, really cool long-term time series. And I think that's the last one I have for phytoplankton. Now moving on to zooplankton. Again, the purpose of these very brief overviews is just to give you a flavor, especially those of you who may be thinking about, who are students who may be thinking about what you want to pursue in the future, just to give you a taste of some of the different types of studies that the FlowCAM has been used for in the past. So as we move on to zooplankton, a really interesting paper by Carnan et al. From 2017 was used to look at copepod size and biovolume. These are from mesozooplankton samples that were preserved in formalin, and comparing different size measurements within the visual spreadsheet or the FlowCAM software compared to traditional microscopy. So in the upper left corner of the figure that I've excerpted here, we can see a direct comparison of biovolume and cubic microns using traditional microscopy compared with the FlowCAM and its 2 different ways of measuring diameter, which are area-based diameter and equivalent spherical diameter. So those of you who are FlowCAM users have probably seen these acronyms before, ABD, ESD, and wondered, okay, which one should I use and why? Because that has huge implications for how you interpret the data, especially when you're looking at size distributions over time. And what the authors of this study found is that area-based diameter was the most comparable with traditional microscopic methods of measuring zooplankton. Equivalent spherical diameter, or ESD, significantly overestimates the volume. And this has mainly to do with the appendages. When we're talking about zooplankton, it's kind of a different beast when you think about it compared to microphytoplankton, because there's oftentimes antennae that need or not be incorporated, and different appendages that are captured in different ways using different techniques. So really interesting paper. They also highlighted, again, how people, that's okay. I just wanted to go back to this point I forgot to mention. They made an interesting comparison between the speed of the FlowCAM and traditional microscopy. So they found on average, they could collect 3,000 specimen in 5 minutes compared to 1 specimen in the same amount of time using the microscope. But remember, like we talked about before, there's benefits and limitations. What you gain in speed, you lose in taxonomic resolution. So there's pros and cons to both FlowCAM and traditional microscopy. But I thought that was cool. You don't too often see authors quantifying the time spent per specimen. And I thought that was really interesting. Okay, move on to the next one. Next, and I think this might be my last zooplankton study, is Haraguchi et al. From 2018. Really interesting study focused on ciliates, which I love. So these images were collected from a fjord in Denmark. And some of the key research questions that the authors were hoping to answer with the FlowCAM and other techniques were whether or not ciliates were the dominant grazers in the community. And if so, what's the proportion of mixotrophic ciliates and how much do they contribute to the transfer of energy to higher trophic levels? This is an excerpt from one of their figures that shows just the diversity of some of the ciliates they saw in their study. And they did find, if I recall correctly, that yeah, ciliates were the dominant grazers. Next, one paper I wanted to highlight that focuses on particle transport. So if we think back to the overview of the biological pump and all these different processes happening in the ocean that people may use the FlowCAM for, sinking aggregates and sinking particulates, this is a really important one. And this study I thought was really, really interesting. The authors were staining Emiliana huxleyi with alcyon blue to assess the production of transparent exopolymer particles, or TEP, and what effect does that production have on the aggregation of particles and ultimately on the sinking of the organisms. They were also looking at the role of viruses. Obviously, you can't look at viruses using the FlowCAM, which hopefully, if you were wondering, I'm sure nobody was, but just to point out, you can't use the FlowCAM for that aspect of the study, but it was an important component nonetheless. So I think that was it for me on, again, just some of the highlighting a few key applications within marine science, a few key studies. There's, like we said earlier, over 300 papers that we know of. I'm sure there are many more. But we just wanted to end, I'm going to turn it back over to Harry to talk about a recent paper, just highlighting the impact of reporting in FlowCAM papers.
Yeah, thanks, Savannah. Yeah, the last paper we're going to talk about, and this is kind of a little lesson for everyone. It is a paper that focused on the FlowCAM, but it's really talking about methods. It was just published in L&O Methods by Owen et al. From Murdoch University in Australia. And what they're talking about, their paper is on, I'm going to read it, reporting of methods for automated devices, a systematic review and recommendations for studies using the FlowCAM for phytoplankton. I'm not going to read I am going to read a little bit of the abstract, just the highlighted parts. And I'm going to read it here, and then we'll talk about it for just a minute or two. So in their abstract, they write, accurate and detailed reporting of methods is essential for scientific progress. Yet it is widely accepted that authors across all scientific fields, we're not just talking phytoplankton and marine science, but authors all across all scientific fields tend to provide insufficient methods detail. Given the recent proliferation of automated and semi-automated technologies for data collection, to address this widespread issue, the details needed for interpretation and reproducibility for each specific technique first need to be identified. A systematic literature review assessed the comprehensiveness of method details reported by 116 peer-reviewed studies published between 2017 and 2020 using the FlowCAM. Through this review and 3 case studies, we identify several key methods that should be reported by FlowCAM studies to ensure their findings are credible, comparable, and replicable. A methods reporting template, and this is at the end of the paper, a methods reported template is presented as a guideline intended to enhance the quality, interpretability, and repeatability of future FlowCAM papers. So what Owen and all are essentially talking about is, hey folks, when you're writing a paper, you're using something that's some new technology, a new method, you really need to really get into the very specifics of your methods. And this is really, this particular study, an exhaustive study. Owen and all, they read 116 studies. We worked with them a little bit over the last couple of years because it took a long time. But notice what they're looking at is, what this particular table is showing, it's a list of all of the 116 papers. And the matrix is showing the different types of the details and the methods and the actual specifics of what are the settings, what kind of FlowCAM did you use, how did you go about your samples. And what they did, they reviewed every single paper and compared all of the methods in each paper to all these different items that you really need to discuss and identify and mention in your paper. And these are just some of them right here. I'm not going to go through every paper, obviously, but it really is a fascinating paper. And again, they chose the FlowCAM because there's been lots of papers published with FlowCAM data to talk about what scientists should be doing when they're discussing how they're using different types of instrumentation. And really what they're showing here is how many papers where the scientists and the authors really didn't get into the details of describing how the FlowCAM was being used, what model, what were the settings, et cetera, et cetera. And I'm just going to jump briefly to Appendix E, where they actually got into many different portions of how a FlowCAM can be used, whether it's the instrument itself, what type of camera is it, what were the objectives that we were using, then how did you set up the FlowCAM? One of the things that we didn't show you today is there's so many different settings that really will influence the way the FlowCAM collects and analyzes data. And the authors are saying, hey, you really need to get into the details of all of this stuff. And then finally, just also how you work with the sample and how you prepare the sample. And Savannah talked about that quite at length earlier. Anyway, a really cool paper, Owen et al. Just was published in L&O. We have this paper. It is open source if anybody would like to read it. But we encourage you to read this paper, whether you're publishing FlowCAM information, IFCB data, CytoBuoy, what have you. It's a really good paper telling you folks all the details you really need to identify and include in your paper. And that, I think, is everything we wanted to talk about.
Yeah, and I realize we're at 11 o'clock our time, Eastern time. I didn't think that we would talk this long. I thought we would have a little bit more time for questions. I have been starting to, what?
Maybe we should just jump into questions.
Yeah, let's go ahead. Or Rubens, I see that there have been questions in the comments or the chat that I've been starting to respond to as Harry was speaking. How would you like to coordinate questions if we have those of you who want to stay on?
Yeah, first of all, thank you so much for the outstanding talk. It was very informative. And I think the audience really took advantage of the experience. It's really great to see how the instrument has improved along the years and how the different applications are open for different communities, including aquatic sciences. I think we could go through the questions first in the chat, those that were not answered yet, and then we can open for additional questions if there are any.
That sounds great.
So please feel free to go through the chat. And I think there's one question regarding having different specimens together in an image. And there's one example here where of a small copepod inside a jellyfish and stuff like that. But I think there are other situations where you have touching organisms or organisms that are somehow in the same ROI. So how the software will deal with that. I think this is a problem not only related to the FlowCAM.
Indeed. Yeah, no, this is an excellent question, and I'm so glad this was brought up. I mean, speaking first and foremost to the initial question, which is if you have an organism that's been ingested by another, you know, one that's inside of another, the FlowCAM is just going to capture that as one region of interest or one vignette, however you choose to say it. So it would count that just once. And if you were to classify that sample, you would have to make some sort of decision as the human operator of where that image should live. So if it were me, you know, perhaps we would have a separate class. And it, again, ultimately depends on what the research question is that you're trying to answer. You could have a separate class that you create manually to kind of isolate those ones that are combining images. It's not ideal, but it's the best thing we have. To the second question about what about, you know, organisms that are not one inside of the other, but one next to the other, a similar situation, you know, once the FlowCAM captures that image, unless you were saving raw camera images, which are raw images of the entire field of view, you can't go back and reprocess or sever the image into two separate ROIs. So it would have to be probably classified in a separate bin. Not an ideal solution. If you did have raw camera images saved, you could reprocess the sample using file processing mode and change what we call the distance to nearest neighbor, which is how far apart do two particles have to be imaged separately. Yeah.
So there's a question, Valentina Torelli, how are you? Thanks for asking the question. Is it possible to share libraries? Yeah, it sort of is. It's something we've worked on a little bit. We haven't gotten too far with that. We have taken libraries that are collected on one FlowCAM, used to apply to data collected on another FlowCAM. They do pretty well. We haven't set up any mechanism where we have some, we've tried to come up with a forum where people can share information, something like a user group, but it hasn't really taken off yet. You know, it's something that's possible, but we're not quite there yet.
Harry, if you may, one alternative for that might be to use Ecotaxa, because
absolutely
you will have your own classification there, and if you have access to different projects using the FlowCAM, you can take advantage of the previously established libraries.
So we're not going to spend we're not going to talk about our new AI platform that we'll have sometime, I hope, in the next 8 months or so, probably not in 2022. But we're hoping that that will allow people, it'll be a whole different way of doing image recognition, not based on statistics, it's all going to be based on imaging. But that's a whole nother discussion. What else? What other questions?
Is it possible, it's from Jamina, forgive me if I'm probably mispronouncing your name. Is it possible to combine in one FlowCAM particle size range from 50 microns all the way to 5 millimeters? In other words, combining data from the FlowCAM 8000 and the macro. The short answer is yes. All the FlowCAMs use the same software, visual spreadsheet. So you could have a run open collected from an 8000 next to a run open on the macro. It is possible to combine individual runs into what we call sets. So that's where basically you're mashing the images into a single file, if you will, within the database. There are some caveats with that, especially when comparing data from different instruments that have vastly different settings. So we would definitely want to talk about that in more detail. So the short answer is yes, you can, but with some limitations and caveats.
So there's a question, Luis. Hey, it's nice to hear from you, asking about a ballpark price for the FlowCAM macro. Actually, we should have mentioned that, and I'll just do it real briefly. Savannah talked about the different models that we have. We have the FlowCAM 5000 with the fixed objective. That's right around 45,000 US dollars. When we get into the FlowCAM 8000, whether you get the 8100 that doesn't have a laser or the 8400 that does have a laser, if you get all 4 objectives, the 8100, no laser, that's right around 85 to 90,000 US dollars, and this would include training. Then when you get the instrument, the 8400 with the laser, we're getting up around 115,000, 120,000 US dollars, again with all 4 objectives, additional software. And then the FlowCAM macro is more around 60,000, 65,000 US dollars. So those are kind of some ballpark pricing on the different instruments. Okay.
Comment from Jenny, not a question, but a comment.
Oh. Thank you, Jenny, for wishing me happiness in my retirement. I'm not going to dwell on this. I've loved doing this. I hope to get to still go to some of my conferences. Rubens, I hope there's another zooplankton conference one of these days. And I still actually hope to be doing some work with Savannah, especially here in Maine with citizen scientists and maybe elsewhere around the world too. So thank you.
I think this might be our last question from Joaquin. Yep. What's the recommended workflow to make images available to users? Custom code? I think there could be a few different angles that he's getting at with this question. Joaquin, feel free to chime in and correct me if I'm mischaracterizing your question. But I think what you're asking is, how can you extract images from visual spreadsheet? What would the workflow look like if you wanted to do your own custom coding, your own, if you wanted to do more advanced machine learning techniques? Okay, great. Thanks for confirming. Right now, it depends on the software version that you have. We do have an application that's been written to extract images from Visual Spreadsheet 5, but it hasn't. Perfect. You have 5. Yeah. Thanks. But it hasn't been fully integrated into the software version that everyone else is using. So we have a tool. We hope to release it with future versions of Visual Spreadsheet 5 and future versions. So, but in the meantime, if that's something you want to work on, you have an acute need for that, send us an email and we can see what we can provide you with in the meantime in between software releases. It's a great question.
Anything else there? Thank you, Joaquin.
Yeah.
Good. Okay.
Oh, we missed. Did we talk about that? Oh, yeah, we did talk about that one. I think if there's any questions that we didn't hit on that anyone is hanging around waiting for us to answer before we sign off, please speak up. I think we've seen them all.
Yeah, I think so too, Savannah. And many users are now leaving the room, so I think it's time to close the session. Okay. And thank you very much again, both of you. Wonderful presentation, for being here with us, and for the audience as well. And I would like to remind all of you who still remain in the room that we'll have a 2-week break now with the course. We come back in 3 weeks from now with a new talk. Okay, I hope to see all of you here. And we will put available the talk online on the Itapina website. As I said at the beginning, that might take a week from now to have this talk available. So please be patient a little bit, but I will announce through the email where the link is, and you will have the opportunity to come back and to the details of this talk and see the videos, for instance, in slow mode. And if you have additional questions, please feel free to send them. You can write me an email, and I will forward the questions to Harry and Savannah.
Thank you.
Thank you very much again, and hope to see you in 3 weeks from now.
Yep. Thank you all. That was a lot of fun. Thank you, Rubens.
Thanks.
Thank
Thanks very much. Cheers.
Ciao, Valentina.
Ciao, Harry. Read the messages for you.
Okay. Thank you.
These are messages.
Yeah.
Oops.
Okay.
Thanks,
Thanks all.
Valentina.